Imagine this. You are stranded on a scorching, deserted island with no food or drinkable water. How would your body react? You're probably thinking that your body would enter a survival state until shelter, food, and water are found. Bacteria function very similar to this, via the stringent control, a stress response that aids bacteria in surviving through environmental stresses. It does this by reducing RNA and protein synthesis when there is a reduced availability of nutrients. Two compounds discovered by researchers Michael Cashel and Jonathan Gallant were shown to mediate this. They theorized that stringent control was directly responsible for controlling a step in energy metabolism. This step would be dubbed the idling reaction, as both researchers postulated that an unknown compound would halt the reaction from progressing whenever tRNA was uncharged and nutrients weren't available. They also stated that the idling reaction would create a phosphorylation inhibitor which indirectly controlled RNA synthesis. A strain of E. coli was grown in an amino acid medium, then separated into different growing conditions. One would be grown in an amino acid mixture, and the other would undergo starvation of amino acids. When viewing an autoradiogram, both strains had 18 visible spots, but a 19th spot was seen in the amino acid deprived strain. This spot was called the magic spot. From performing another test, the spot was shown to be composed of two compounds, MS1 and 2. So what were these MS compounds? And how did it relate to the idling reaction? From testing uracil starvation, they discovered that the MS compounds were not produced directly by amino acid deprivation. So, were the compounds induced by the idling reaction? As mentioned before, the idling reaction was some step not known at the time in protein synthesis that was hindered. The works of Neidhart showed that the idling reaction was the transfer of amino acid to the tRNA of the growing peptide chain via the use of GTP. Amino acid synthetases attach amino acids to its cognate tRNA to form charged tRNA. These charged tRNA were responsible for carrying out RNA synthesis. So, the lack of charged tRNA meant that this step in protein synthesis would not be carried out, indirectly inhibiting the rate of RNA synthesis. This information led Cashel and Gallant to confirm that the MS compounds were the phosphorylation inhibitors they were searching for that was produced by the idling reaction. They classified MS1 and 2 as guanosine tetra and pentaphosphate and grouped them as alarmone. So that gives rise to many questions. Firstly, what produces and controls alarmone levels? The answer is RSH, short form for rel-A SpoT homolog. Under stress, RSH produces alarmone from ATP, GTP, and GDP. RSH is composed of the long RS homolog and small alarmone synthetases along with hydrolase. The long RSH includes the enzymes rel-A, SpoT, and rel. They, along with small alarmone synthetases, are responsible for synthesizing alarmone. Rel-A detects the levels of amino acids by hopping from ribosomes. If uncharged tRNA is present and enters the A site, it means there is a deficiency of amino acids. Rel-A will then deattach and activate the synthesis of alarmone. SpoT balances the concentration of alarmone. In nutrient-rich conditions, GTPase promotes degradation of alarmone. But during starvation, acyl carrier proteins activate SpoT to produce more alarmone. The last component, rel, produces and degrades alarmone depending on whether uncharged tRNA enters the A site. Hydrolases, on the other hand, degrade alarmone into GTP and GDP with the presence of manganese and nutrient-rich environments. So, we know how alarmone levels are being controlled, but how does it perform its role? Alarmone does this by binding to two sites on RNA polymerase. The first is between the omega and beta prime subunits, and the second is between the beta prime and transcription factor DKSA. Alarmone allows sigma factor S and DKSA to bind to the RNA polymerase instead of sigma factor D. Sigma factors are multi-domain bacterial subunits needed for the recognition of the RNA polymerase onto the promoter to control which set of genes are transcribed. The difference between both mentioned sigma factors is that sigma S facilitates the transcription of starvation response genes, while sigma D facilitates the transcription of maintenance genes. When alarmone and DKSA binds to the site, it causes the increase in the transcription of amino acid biogenesis genes. Thus, alarmone functions by allowing stress response genes to be encoded instead of other genes by targeting RNA polymerase. After decades from their initial discovery, there is still so much more to learn about Alarmone. Recent studies are viewing its effects on bacterial pathogenicity and persistence. In addition to this, a third Alarmone has been discovered, being dubbed as PGPP. But we'll leave these stories for another day, possibly for if we ever get stranded on a deserted island with a lack of food or water and maybe have nothing else to talk about.